Um, so we'll start with the bad news. I do not have your tests finished yet. But the good news is I will have them. They'll be posted later tonight, maybe, or first thing tomorrow morning. Yeah, I like that. Yeah. I see a couple of shocked faces. That is true. I overestimated how long it would take me to do something else. So. But the ones I have graded, and I've been going in a random order, just picking and choosing. The ones I've graded have been good. So I think most people will be happy with their grade tomorrow or later today if it were. All right, which feels weird. So because we were
recording in progress.
It's for everyone. It might come out that way. I don't know. The grades have looked good. You all maybe have come close to earning your own A's, which is I always say, I don't know if you ever heard this, and I thought this when I was in college. I don't remember grad school, but it was the idea that professors wanted students to do not necessarily poorly, but they wanted their exams to be viewed as like really hard. And at some point, a professor I had said, "Why would I want everybody to fail my exam? That means I didn't teach them very well." I thought, no, it's definitely a balance between you all performing well, but also, the professor's got to teach, got to write a good exam, or at least a fair exam. So I had a biochemistry teacher. I ended up having him twice because I dropped it the first time because I went to the first exam, I sat down, I looked at the exam and thought, I have not studied enough. So I just stood right back up, turned in the blank exam and said, I'll see you in a year when you offer this class again. And so the way he did his exams, he expected that the average would be, in the upper 40s or 50. And so that would be that would kind of fall at the C plus B minus range. And so what he did is his grading scale for the entire course, like your final grade. I think I got an A when I actually took it and finished it. I got an A, but I think I had like a 79 in the class. But the way he adjusted his scale, it was like 75 and up was an A, and that's tough to handle. So I try to not do that. Okay. Enough rambling. So you will have exam 2 back soon, but we are also at the point now where we are doing the final chapter that we're going to cover for exam 3. And exam 3, I believe, and somebody can correct me, exam 3 will open up next weekend. I think that sounds right. So we're a little bit away from that, and then it'll be built the same way. So same style. All right. So last chapter. We have got our last chapter for this unit, and actually this represents the last unit that we are covering things that are not related to a disease directly. You'll see units 4 and 5 are different collections of types of diseases related to the immune system. And so what we'll hit in the next unit, we'll still cover some of this basic information, and all of the stuff we've talked about, you'll get to see applied to a particular disease. So we'll do hypersensitivities and autoimmunity. We'll cover that first, and then we'll follow up in the last unit for our class with transplantation, infectious disease, and time permitting, and I think we'll be okay, cancer biology. So lots of interesting stuff. Also, in the next time we meet, 6 of you will present your papers. So make sure you have your PowerPoint. Now for these first ones, as long as it's submitted before class meets, that's good with me because that allows me to load them if I need to load them. For those that are scheduled to go on Thursday, if you can, maybe set an optimistic goal, have it done by Tuesday and loaded, or at least know that downstream, we might have some people go a class period earlier than they're scheduled for, although in practice, that's probably going to be unlikely, but I'd rather you be prepared than not. So we're over halfway. Let's get started. Halfway with the course. Let's do it. So a brief overview. We're talking about T cell activation now. We had a separate chapter, the chapter preceding this, where we talked about the biology of a T cell. What things is it expressing? How is it developing? And then what are the sequence of events that are going to activate it? So what we're looking at now is we're talking about naive T cells, which recall naive T cells are mature, but they have not yet seen their specific antigen. And so these naive T cells, to find their antigen, they circulate around, they move through secondary lymphoid tissues. If they don't see anything, they go back to the blood. And when they do see their antigen, they have a series of events that occur that results in their activation. And you will either get, a large majority will be effector T cells. And these are the ones that go and do the things, right? They secrete cytokines or destroy the cells. Much of what predominantly what we're talking about today are the consequences of effector T cell activation. And the other thing they'll generate are those memory T cells. And because T cells are a part of the adaptive immune response, they recur. And so these memory T cells can react more quickly, generate a faster response upon second exposure to the same antigen. Now, the most commonly activated T cell types fall into one of our two categories. We've got our CD4 positive T cells, which you might also see called T helper cells or TH, variety of types. We'll talk about the subsets here. And ultimately, CD4 positive T cells, when activated, produce cytokines and signals that guide responses. In CD8 positive T cells, when they get activated, we might also call those cytotoxic T cells. And these are the ones that are going to go, and when they see a cell that is expressing the right thing, it's going to kill that cell. And so it results in target cell lysis. Now, T cells follow a 2 activation signal. And what will end up happening is we're going to say they have 2 signals, but then they have an air quotes third signal that is intuitive. You know this is going to happen. We're now just giving it I usually put it in quotes and italicize it. First signal, we already know the antigen has to be observed. So the TCR on T cell needs to interact with a specific peptide that is expressed in the right kind of MHC. The co-receptors on the T cell, and those can only be either CD4 or CD8. So we don't have any double positive T cells for our purposes circulating through. Recall CD4 will interact with antigen that is a part of MHC2 because half of 4 is 2. And CD8 will interact with antigens being presented on MHC class 1. Now, once we have antigen interaction, we will start to get CD3 activation. CD3 is our signaling molecule. Co-receptor activation makes that even stronger. It lowers the threshold required to activate that T cell, and we get a series of intracellular signaling events. Now, as that happens, to get a T cell completely activated, we need not only this first signal that's antigen specific, we also need a second signal, which is not antigen specific, and this is the co-stimulation. And so we have the interaction of these co-stimulatory pairs, where a molecule on the T cell is interacting with its ligand on the antigen presenting cell. And what this does is it results in intracellular signaling events, and we take these two together, we get complete activation, which ultimately leads to the production of materials that allow these T cells to survive, proliferate, and ultimately differentiate towards this antigen-specific response. So if you like the pictures, here's a good place to remind yourself what these are. The first signal is related to antigen interaction, and who are the molecules involved in that. And then we have co-stimulation. And so recall, we have a variety of different types that we will revisit here. And many of these are consistent, right? B and T cell activation, although not identical, is homologous, right? The nature of interactions are kind of familiar between the two. Now, a brief word, dendritic cells. We talk about dendritic cells because what we're going to describe when we talk about how T cells get activated really is we focus on CD4 positive T cells, and we do that because there's more diversity in their activation responses. But also, when you learn the activation pattern for CD4, you have then also learned nearly all of the activation events required for CD8 positive T cells. So it's a good excuse to go ahead and talk about dendritic cells. And dendritic cells are a type of antigen presenting cell, or are a type of professional antigen presenting cell, meaning they express MHC class 2. Recall, we need MHC class 2 because we're talking about activating CD4 positive T cells. Dendritic cells have the ability to ingest antigen and process it. And so what they do is they take in these protein antigens and they chew them up so that they can show them and recall, why does it have to be processed? And also, why does it have to be presented in a linear fragment? That's because that's how T cell receptors are able to see that antigen. And so dendritic cells have these characteristics, and they tend to be the primary, or at least the antigen presenting cell we will refer back to most when we're talking about activating our T cells. There are two major subtypes. We referred to one of these by name before, and those are the plasmacytoid dendritic cells. And plasmacytoid dendritic cells are actually, they come from the lymphoid lineage, but they seem to play big roles in antiviral responses because once they're activated, they secrete large amounts of interferons, which drive antiviral innate immune responses. We compare those to myeloid dendritic cells, and these develop out of the myeloid lineage. When we talk about dendritic cells, in most contexts, what we mean are the myeloid-derived dendritic cells. If we're talking about presenting antigen to T cells, for our purposes, we mean myeloid. So henceforth, you will just see me call those dendritic cells, but know that there are two varieties. And these myeloid dendritic cells, because of their ability to capture and express antigen and secrete the right things to activate T cells, they induce T cell responses. Now, dendritic cells can be found in circulation. You can find dendritic cells bumping around. They may be better characterized as being found within tissues. They're especially prominent at areas where you have antigens that may be on their way in. So think we have dendritic cells related to the skin and the intestinal tract and the respiratory tract. All of these barrier surfaces, there will be dendritic cells in the area. You also have dendritic cells that populate secondary lymphoid tissues, such as lymph nodes. You even have, if you think back to the previous chapter, we talked about dendritic cells that are found in the thymus. You know, what are those dendritic cells doing? They're helping with the selection process to know which T cells survive and which ones don't. So we can find them in tissues, barrier, but also lymphoid. Now, a key feature of dendritic cells, besides their ability to present antigens, they also can be migratory. And really, depending on where a dendritic cell sees antigen, will tell you whether it's one that can move or not. So dendritic cells that are within non-lymphoid tissues, such as the skin, would be classified as migratory, which means when they capture that antigen, they leave the place that they captured the antigen, they work their way into the lymphatics, and they go to what is called the draining lymph node. And that is the lymph node that is responsible for draining lymph from a particular region. And so Langerhans cells are dendritic cells related to the skin. So if on the tip of my finger, a Langerhans cell captures some antigen, it gets the right stimulation to say, I can leave now. What it'll do is it'll leave and migrate to the nearest lymph node that drains through the lymphatics. And when it gets there, what it's doing is it's reporting the antigen it's seen to the cells of the lymph node. Recall, that's like, how is it that the adaptive immune response can take all of the possible antigens that maybe diffuse throughout the organism and put them in a position where they can be seen. We drain our tissue fluid through lymphatics where antigen gets concentrated, but also where lymphocytes move through. Now, dendritic cells that are present in lymphoid tissues are considered non-migratory. So they'll still get antigen, but they don't really need to go anywhere because they are already there. And so you also see these called lymphoid organ resident dendritic cells. Now, dendritic cells can need to be activated. Immature dendritic cells, which is illustrated here kind of on the top panel, or I guess in this top upper left. Immature dendritic cells have not yet encountered an antigen. So what this means is they've not seen any antigen, so there's not really any threat yet. And so if you look at their surface expression, they express low amounts of MHC class 2. Because they haven't seen antigen, they don't have anything to show. But what immature dendritic cells do, they are an innate cell, right? They're an antigen presenting cell. So they express many of our pattern recognition receptors. And harken back to chapter 2, what are those? Toll-like receptors are especially prevalent. C-type, C-lectin type receptors, those are especially important. And so we have these structures that are able to bind those PAMPs, these things that are more related to a bad guy. They're able to interact with those, and they also express phagocytic receptors so that these immature dendritic cells can see pathogens or possible pathogens with their pattern recognition receptors, but they can also capture things with their phagocytic receptors. And when they encounter antigen and when they capture this antigen, what that does is it stimulates antigen processing within the dendritic cell. And what this does is it activates it. So now we have a mature dendritic cell. And these are mature because they have captured antigen and have started processing it. Mature dendritic cells start to change what they're expressing. Because a mature dendritic cell needs to show that antigen to activate a T cell. And so what it does is it upregulates MHC class 2, because remember, it's captured antigen, it's processing it. So now it's going to show it, especially with MHC class 2. It will begin to express co-stimulatory binding partners. Recall, T cells and the antigen presenting cell need to interact. And one of those interactions is through co-stimulation. And the B7 family especially interacts with the T cell molecule called CD28. And importantly, a mature dendritic cell, once it's undergone this maturation process, will start to produce cytokines that help drive the response. And one of the more important ones is IL-2. And without looking, you should know at this, or one of the things you'll want to make sure you know if you don't already, IL-2 drives lymphocyte proliferation. So T cells in the presence of IL-2 are getting a let's live and let's make more of us signal. So now we have a mature dendritic cell that can show antigen, it can do the co-stimulation, and it's secreting the stuff to help the cells it's trying to activate grow. In addition, our dendritic cells that are out in the tissue that are migratory, when they get activated, they're able to change some of their chemokine receptors. So they express things like CCR7, which allows them to leave the tissue and migrate towards the chemokines that the high endothelial venules are expressing, so that the dendritic cell can leave the tissue and carry with it the antigens it's presenting to go into the lymph node. All right, so now it's in the place where all of the T cells are circulating through, because what it wants to do is it wants to show its antigen to those naive T cells. Now, the course of this interaction between a dendritic cell and a naive T cell, really you need that interaction, or that interaction occurs within about 8 to 10 hours of a dendritic cell seeing antigen. And we can track this in especially animal models where you can label them and you can watch the dendritic cell moving through the lymphatics. And the idea of having dendritic cells in an immature state and then having them mature once they see antigen really helps to block or slow down any self-reactivity. Immature dendritic cells express low levels of MHC class 2. And what this means is they can only start to show antigen when they get a sufficient enough response, such as seeing something that may be a threat through your pattern recognition receptors. Turns out but we could find those DAMPs, right? But most self-antigens that a dendritic cell may encounter are not terribly good stimulators of an innate immune response. This idea of getting dendritic cells more active and maturing is also one of the reasons some vaccines have an adjuvant, right? The adjuvant provokes a just generalized inflammatory response, and that makes dendritic cells behave better so that the antigen you are also giving in the vaccine maybe gets processed better and presented better, and you get that immune response to it. And so to activate CD4 positive T cells, this is a fantastic little snapshot. There's a similar one that I realize I left out. It's in your text, but I didn't put it at the end of chapter 10's PowerPoint like I did with B cells in chapter 8. But this gives a really good illustration of who is interacting with whom. right? So naive CD4 positive T cells need to see antigen that's being presented on antigen presenting cells. Principally, these are going to be dendritic cells, but just somebody who can show antigen. And because these are CD4 positive T cells, they have to see that antigen, that peptide complex with MHC class 2. What we're going to do in the next series, number of slides, is just look at the summary of these interactions, much of which we've already gone over, but the redundancy and the repetition is good. So the peptide that is in our MHC class 2 has to get with the TCR. And we're talking about alpha beta TCR. Make sure you know that you are able to distinguish the difference between those from the last chapter. This interaction represents the first signal. The antigen presenting cells going to have that MHC class 2 loaded with processed peptide antigen, and it's going to interact with the variable regions on the alpha and beta chains of our TCR. But to get sufficient activation, we need time. And so there are other interactions that help stabilize that cell-to-cell contact, as well as increasing the duration of interaction, which improves signaling, because when a T cell is interacting with an antigen presenting cell to get activation, it looks like multiple hours are needed. And so you want those cells to be able to hang tight together. Now, as a part of this first signal is also the interaction that occurs with MHC and the T cell co-receptor. And in this case, it's MHC class 2 and its interactions with CD4. Again, this enhances the signal. It increases the stability of the two cells interacting. And what this does is it lowers the threshold for that T cell's activation by 100 times. So you also need a second signal. So the first signal is seeing antigen and interacting with the co-receptor. The second is our co-stimulation. And this, again, helps to provide the second signal. And it's a collection of interactions. And we have two highlighted here. We have the interactions between the B7 family of receptors that are found on the antigen presenting cell. And these are collectively CD80 and 86. And they're going to interact with CD28, which is the co-stimulatory molecule on T cells, and they're going to interact. And when that interaction is successful, that will initiate an intracellular response that gives you complete activation of the T cell. The longer T cells are activated, what will end up happening is that they will start to express another molecule that is similar to CD28 called CTLA4. And you'll see that they are showing dual interactions. CTLA4 binds to B7 members on the antigen presenting cell much more tightly, which actually prevents CD28 from engaging. And for a couple of reasons, it ends up turning off the T cell. So CTLA4 is a negative signal. The other co-stimulatory pair is CD40, which interacts with CD40 ligand or CD40L. And what this co-stimulatory molecule or co-stimulatory pair does is it enhances the co-stimulation that's occurring by our B7 family with CD28. When CD40, which is presented on our antigen presenting cell, interacts with CD40 ligand, which is presented on our T cell, it stimulates or it improves both binding partners. But one of the things it does is it causes the antigen presenting cell to express more B7 family members, which makes co-stimulation more likely. There are also going to be adhesion molecule interactions, because recall, we not only need to hold these two cells together, but lymphocytes are moving through the tissue. And so if they encounter an antigen presenting cell that has an antigen they want, they need something that can help it to slow down, stop moving, or just interact and hold together. So these are a collection of molecules, and we referenced 3 pairs. We're just going to reference these again. And the whole net effect of these adhesion molecules is to increase strength of the cell-to-cell contact, make this interaction more stable, because really what we need to do is increase antigen scan time. And that's the time that it takes a T cell to interact with a possible peptide. And so the longer that can occur, the more likely you can have the appropriate binding, the specific binding that's required. Now, the major pairs of these just illustrated here on the bottom of the figure, and I've just typed them back out just so that they're typed. ICAM1, which is on, so all of our first members are antigen presenting cells, antigen presenting cell molecules. So ICAM1 will bind to a T cell molecule called LFA1. LFA3 on an antigen presenting cell will interact with a T cell molecule called CD2. LFA1 on an antigen presenting cell can interact with ICAM3 on a T cell. And so all of these are going to be occurring, and that's going to really hold those cells together. But also, if a T cell is passing, it might slow down, bind it, and say, oh, this is expressing something I like. Now, those events, those events taken together are antigen recognition. And when that happens, we're going to stimulate an intracellular signaling cascade. I would imagine you've talked about this again, but if nothing else, think back to cell biology. We see something on the outside of the cell. Well, how is it that we take this thing that's on the outside and we cause the cell to change its behavior on the inside? And so we're going to depict this with CD4 positive T cells because that is what we are covering at this moment. But essentially, the intracellular signaling events for CD8 positive T cell activation, for our purposes, identical. Oh, and also these signaling events are not, while not specifically identical, are very, very similar to B cell activation, right? And so we again see some redundancy and some repetition. And so it's important to be able to place which molecule is related to B cells and which molecule is related to T cells. Being able to organize them is nice. So I've put this on its own slide because I think this is how, for signaling events like this, I would turn to the schematic. Words are fine. I would also use those from your text and from my notes. But this figure just shows it nicely. And so one of the things, if you're able to replicate this figure, you can draw it out, you can trace the events. You are, 75, 2/3 of the way there to knowing these signaling events inside the cell. So I put that there at the beginning as a place to refer back to, but you'll see on all of the following slides, I have it placed when necessary. Now, I know we referenced this in chapter 10, but there is this synapse that forms. And this is when, and it's called the immunological synapse because it is the space where a T cell is interacting with the antigen presenting cell. And just like you have the synapse with neurons, which is where a presynaptic neuron is interacting with a postsynaptic neuron, we have this area of contact. And so there are some differences between neurons, but it's an intimate relationship with a very small space between. So this is where an antigen presenting cell and a T cell are interacting. So what ends up stimulating the formation of this immunological synapse is actually the interaction between T cell receptor and the peptide and MHC class II. This synapse forms, and if you start to look, what we are seeing is here are some adhesion molecules, and here are co-receptors. This synapse forms because you have multiple things anchoring these two cells together. And what this does is it allows for our signaling molecules inside the T cell to aggregate and to cluster very easily. Principally, we need that CD3, which is always affiliated with the T cell receptor. We want to make sure that the things that need to interact with CD3 are there when they need to be. And so this synapse and just membranes in general are very dynamic. So at any point, the immunological synapse may be different because it depends on the events occurring relative to interaction. So how do we do this? Cells use lipid rafts, and these are discrete units that kind of move together. Clusters of proteins that all belong to the same process will be housed together and can be, for example, the lipid raft can be moved to the immunological synapse when it's needed, and then things that aren't needed can be pushed away from the immunological synapse, so that the only things that are present are the things that are needed to get good activation of that cell. Now, the intracellular events, we again need that initial signal, and that is when our T cell receptor, it's the variable portions of the T cell receptor. And again, we are interacting with peptide and MHC class II. So this T cell receptor has to be alpha and beta. Now, likely this initial signal that makes ultimately CD3 get activated is probably because we either get a change in the T cell receptor, which makes available CD3s, the regions of CD3 that can get activated, or you just get enough T cell receptor clustered together that's all being bound, and it activates it. Either way, CD3 gets made available for activation. What we have forming now, or what we have the ability to do now, are form intracellular signaling complexes, because when CD3 is available for activation, it gets activated by kinases. And there are specific regions where it gets activated. And you can see these are drawn with rectangles. And these are called ITAMs. And the A in ITAMs mean activating. So kinases are enzymes that phosphorylate things. And so these ITAMs get phosphorylated. And what that does is it allows for the recruitment and the binding of the next step of the signaling pathway. So when you activate the T cell receptor directly, you get the recruitment of a kinase called FIN or FYN. When you activate the co-receptor, CD4 in this case, you're going to get the recruitment of a kinase called LCK. And these two together are going to they're going to work together to recruit and then activate a protein called ZAP70. And what ZAP70 does is gets activated by these these kinases activate CD3. I had that backwards. And that allows ZAP70 to bind to these now activated ITAM sites. And it turns out that ZAP70 is absolutely critical for T cell activation. Deficiency of ZAP70 means no T cell activation or no T cell responses. And so you can see, why is it that co-receptor engagement enhances a signal? Well, because if the co-receptor is engaged, it activates an additional kinase that can activate CD3. Now, once ZAP70 is bound, it gets activated by that same LCK kinase. And when ZAP70 has bound and is activated, and when we say activated, you can always just assume a phosphate group is added. When ZAP70 is activated, it then binds an adapter protein called LAT. And what LAT does is it allows the binding of a collection of molecules, one of which we're going to follow. It allows the binding of SLP76 and then the binding of an enzyme called PLC gamma 1, phospholipase C gamma 1. Now, the one we're going to follow is PLC gamma 1. PLC gamma 1 recruitment. It's now recruited. It is now in a state that it can be activated because all of these preceding events have occurred. And so what does it do? It initiates pathways that ultimately lead to the production of 3 transcription factors. And recall, transcription factors enter the nucleus and stimulate transcription and expression of specific genes. So we're going to follow the pathway that leads to the transcription factor NFAT. PLC gamma 1. It's been activated. All of the preceding events have occurred. And what it does is it cleaves a membrane phospholipid called PIP2. And that cleavage by this enzyme generates 2 products. One is a soluble product called IP3, and the other is a membrane-bound product called DAG. So for NFAT transcription factor, we're going to follow the soluble piece, which is IP3. And when that gets released as a result of that enzyme's cleaving that phospholipid, it actually stimulates the release of calcium into the cytosol. So normally cells keep their calcium sequestered. IP3 is going to cause that calcium to flood in. And when that occurs, it activates a protein called calcineurin. And what calcineurin does is calcineurin leads to the activation of the transcription factor NFAT, which can then enter the nucleus. The next 2 pathways are a little bit smaller, so I just included them on the same slide. We're going to follow the NF-kappa B transcription factor. And this is a result still from the activity of phospholipase C gamma 1. And that is when it cleaves PIP, it generates IP3, but it also generates a membrane-bound DAG. And what DAG can do is it can activate protein kinase C, calmodulin, and calcineurin, utilized for different purposes. Yeah, the one we will worry with is calcineurin. Somebody asked, what's the difference between calmodulin and calcineurin? So DAG activates protein kinase C or PKC, and what this does is it leads to the activation of a kinase cascade, ultimately resulting in the activation of MNF kappa B. That activates, goes into the nucleus, does its thing. The last pathway that PLC gamma 1 does is it activates the AP1 transcription factor. And here we again are going to see DAG doing something. What DAG does is it allows the recruitment of a GTPase called RAS. And because it's a GTPase, what it does is it cleaves GTP to transfer a phosphate group. Right? It cleaves that and then it resets. So it's constantly cycling on and off. Activation of RAS leads to the activation of the MAP kinase cascade, which ultimately results in our AP1 transcription factor. Now, those three transcription factors, we're just going to take them together. NF kappa B, AP1, and NFAT. They enter, once they're activated, they're going to enter the nucleus. They're going to bind to regulatory parts, regulatory sequences of our genes, and they induce production of our response-specific mediators. And this includes transcription of IL-2. Remember, IL-2 is a cytokine that drives T cell survival and proliferation, but it'll also stimulate production of the alpha chain of IL-2 receptor. So by producing the cytokine IL-2, this is a T cell growth factor, proliferation factor. And by making the alpha chain of the IL-2 receptor, that actually makes IL-2 receptor high affinity. So kind of think back to chapter 12, right? Resting T cells express some parts of the IL-2 receptor, but they're not very good binders. And it's only when they're activated that they add that third part that now makes the IL-2 receptor super high affinity. And when that occurs, and you can see that here, right? And at rest, intermediate, not a terribly good binder, but when you activate and express the alpha chain, it becomes a high affinity binding partner with a very low dissociation constant, which means binds tight. Now, the consequence of that is we have an activated T cell that's producing a cytokine that it needs, and that IL-2 can act on a receptor that it is also expressing. So we get lots of autocrine interaction, which will cause the cell to start proliferating. But what it will also do is that IL-2 can also stimulate the cells that are, it can be a positive message to the cells that are being produced. So what we do is we move towards the production of our interferon, of our effector cells. Now, so that's the events that occur from antigen interaction and co-receptor binding, right? What then of the co-stimulatory molecules? So taking B7, which is on the antigen presenting cell, and CD28, which is expressed on the T cell. When you have that engagement, what this does is it enhances activation to the point that it's required for complete T cell activation. It recruits a kinase called PI3K and increases its activity. So PI3 kinase actually goes and stimulates activation of these transcription factors. But in addition to that, CD28 engagement helps to mobilize lipid rafts that help to bring more of these activating molecules into the synapse. Net result of this is we're going to get an increase in the activation of our transcription factor pathways, which means we're going to get more IL-2, good high affinity IL-2 receptor. And this co-stimulation is what helps to avoid cell death, keeps these emerging T cells from dying, and it promotes their survival and their proliferation. Now, how that proliferation occurs, it can be both autocrine signaling, which is the loop. Autocrine recall is when a cell produces a cytokine that acts on itself, but it can also be paracrine signaling. Now, this paracrine signaling, this IL-2 that's going to drive this proliferation, can be produced by the activated T cell, but recall the events we talked about with our dendritic cells. When a dendritic cell matures, it is capable of producing IL-2, which can help drive this positive T cell response. So we have these proliferating T cells, and they're going to undergo a differentiation process. They're going to become effector cells. Now, we are talking about CD4 T cell activation. Those are going to be the ones that are going to lead to robust cytokine producing T cells. But don't forget, we have CD positive 8 events that are very similar. Intracellularly, they're the same. And extracellularly, there's just a couple of differences, but they become cytotoxic. Now, where these effector cells go, because once they're activated and they start to differentiate, they're going to leave. They're They're going to leave the lymph node where they're being activated and produced so that they can go to the areas to go and fight the bad guys that are activating them. And there are different homing receptors that get expressed depending on where the cell is going. If you're from one of, I think it was B cells, we talked about when they traffic back to the lymph nodes related to the mucosa, they use expression of alpha-4 and beta-7 and CCR9. T cells would do the same thing. And recall, homing receptors are not necessarily cell type specific, but instead are like addresses. Skin would see an increase in CCR10 and CLA, and sites of inflammation, and tissues, with the exception of skin, use a couple of different ones too. Now, this is all well and good, robust immune responses, but we know that immune responses that continue are no good, because what happens is a little inflammation is good, but inflammation that lasts too long is bad. And so we have to control these. Now, there are a few different ways this gets controlled. We're going to talk about how molecules are produced in response that inactivate the message, that turn the response down. You can do this to inhibit cell cycle. And then there's the concept called T cell exhaustion that we'll come back and touch on again. And the concept of T cell exhaustion is that T cells that are being repeatedly stimulated to proliferate eventually lose the ability to have this proliferative effector function. So one of the things that helps terminate it, maybe one of the best characterized, is CTLA4. Recall, it's structurally similar to CD28. And what happens is when a T cell gets activated, CTLA4 expression slowly starts to increase so that you actually see this production about 24 to 48 hours. So it gives the T cell time to activate and stay activated, and then it starts to produce this inhibitory molecule. CTLA4 has multiple effects. It is a direct competitor, as we see here. CTLA4 is a better binder of the B7 family members, CD80 and 86. And so it binds them with higher affinity. And what that does is it leads to the fact that CD28 is not engaging, that you lose a positive co-stimulatory signal. CTLA4 itself is also inhibitory, because CTLA4, instead of having ITAMs with an A, and the A stands for activating, instead it has ITIMs, and it's an I for inhibitory. And what happens when you activate ITIMs, it brings together a collection of enzymes called phosphatases. Phosphatases remove phosphate groups, and from your intracellular signaling cascade, really adding a phosphate group is what turns it on. And so CTLA4 allows for all of those phosphates to be popped off. The signal gets turned down. Additionally, CTLA4 just gets in the way. It just interferes and displaces molecules in the immunological synapse, worsening the connection, making it less stable, making it less durable. And the net effect of CTLA4 is you turn that T cell off so that you get less IL-2 production, which means less T cell proliferation. Now, there's another response inhibitor called programmed cell death protein 1. So we'll just call it PD1. What PD1 is it is also expressed on activated T cells. And it's associated with this concept of T cell exhaustion. So when a ligand binds, so T cells expressing PD1, when it binds its partner on the antigen presenting cell, what it does is it inhibits the function of ZAP70, which is critical to the intracellular cascade continuing. There are different ligands. There's PDL1, PD1 like PD ligand 1, which you find on most tumors and many normal cells. And then you have PD ligand 2, which is predominantly on antigen presenting cells such as dendritic cells, and is also expressed by many tumors. And what you're seeing is that tumors seem to express this thing that helps turn immune responses off. And so you could, in a tumor, try to block that, right? You prevent the interaction between PD1 and its ligand, and that'll keep your T cells happy longer. PD1 interactions can also be viewed in the other direction. If you have T cells that are inappropriately activated, such as an autoimmunity, maybe we want to engage PD1 so that we can turn those inappropriately active T cells off. So there are also alternative methods. What we have looked at is the idea of antigen epitopes, but pathogens and the things we encounter are diverse. Now, when we interact with these antigen epitopes, recall our antigens from our pathogens, recall they're going to have a variety of epitopes, these little pieces that can be interacted with a specific T cell. And that causes clonal activation. Well, there are some agents that induce the activation of multiple T cell clones in alternative ways. And we're going to talk about super antigens and T cell activators. And at least super antigens we've referred to before. So a super antigen is mostly a collection of bacterial toxins that are generated and produced by pathogenic organisms. We referred to the toxic shock toxin that is produced by Staphylococcus. I think we referred to staphylococcal enterotoxin. Essentially what a super antigen does, and I think this picture shows it very well, is it does not matter what that peptide is. Instead, the super antigen can bind the variable region of our beta chain on our T cell receptor. So it binds the T cell receptor, but it can also just bind to the MHC. So the TCR does not need to see the correct peptide. Super antigen forces the two to interact. And what that does is means we're getting TCR activation independent of whether it's recognizing its appropriate antigen. And as you might expect, if you are just arbitrarily activating T cells that are not antigen specific, because you are forcing them to get their messages, we're going to get lots of massive and inappropriate activation that can lead to massive cytokine release and the manifestations of exposure to bacterial toxins. And we can also look at these from another look at this alternative activation in another concept, and that's in the form of T cell activators. As their name supplies, these are things that can activate any clone of a T cell. So these again are independent of antigen. Now we refer to something called a mitogen, and a mitogen is we used that term before, and that's a molecule that can induce T cells to proliferate. But what they do is they induce T cells to proliferate non-specifically. We have some examples that bind to the TCR, but they don't bind at the antigen site, right? They're just stimulating TCRs to the point where they can activate. Some that are used are things like concavalin A or ConA, phytohemagglutinin or PHA, and then a plant protein plant structure called pokeweed mitogen. Now you can also do this, there's an experimental reason or a clinical reason where you might try to activate T cells non-specifically, so to speak. You can use anti-CD3 antibodies, right? Antibodies bind specifically. If you have one that binds to CD3, you give it, it'll bind the part of the T cell that will activate CD3. Wham, you get T cell activation. One used for research purposes a lot is PMA and inomycin. And what PMA and inomycin do is these things are not interacting with the T cell, but instead they're not interacting with the T cell receptor, but instead they're stimulating the intracellular signaling cascade. And PMA and inomycin activate PKC and increase cytosolic calcium, which gives us our signal. So we've got our T cells activated, our CD4 positive T cells activated. What are they going to do? We have those, the bulk of what we'll talk about are how they secrete cytokines, but in that function, they can also help B cells produce good, strong antibody responses. So for our cytokine secretion, activation of CD4 positive T cells is going to give us the production of cytokines. This is when you produce cytokines, those cytokines can interact with anybody that can see them. So we're not necessarily producing antigen-specific effectors, and instead what we're doing is producing things that will cause B cells or other T cells or innate cells or bone marrow to have a response that will that is built to remedy this antigen that's activating it. Now, the general model of differentiation is that we need naive T cells to be activated. As a part of that, we know that cytokines have to be present. And so kind of the third signal of activation includes this ability of T cells to differentiate into different subsets of molecules. IL-2 is one of those cytokines that can be referred to as one of these third signals, right? T cells have to have IL-2 being produced so that they can proliferate. So just having cytokines present will guide which direction we're going to go. What dictates which cytokines we have? Well, it depends on what kind of cells and their responses have occurred as a result of the antigen. Viral antigens will evoke a different kind of cytokine profile, which will drive naive T cell activation towards one path. Bacterial antigens can drive it towards a different one. Now, once we have this influence, once we have driven our CD4 positive T cells towards a particular subtype, they're going to be associated with a characteristic group of cytokines and group of molecules. And what we call these are signature cytokines. And really what we're talking about is a Th1 cell is a T cell that has been differentiated in such a way that it produces a particular group of cytokines. Do know that there's lots of fluidity between, there's lots of overlap between these. You will see, and there's not everybody agrees with what is going to be produced. So what we are looking at is really the hallmark cytokine. Right? We're not going to learn all of the cytokines that get produced by a Th1 cell. We're going to focus on the ones that are most prominent and are best associated with T helper 1 activity. Before we do that, let's refresh ourselves on what it means to be a T helper 0 cell, which was a concept we introduced, I think, last chapter or maybe in one of the previous units. But a T helper 0 cell could either be a T cell that has just been activated but is not yet differentiated. Some texts, some researchers will consider a T helper 0 cell just a naive T cell. Either way, it's one that has not differentiated towards a particular subset. Where that T helper 0 is going to go once it gets activated depends on the cytokine signals that are present. So with that in mind, T helper 1 cells are going to develop in the presence of IL-12. We've seen IL-12, right? It's made by mature dendritic cells. It also gets produced by NK cells. And especially if you see NK cells, you think altered self. So what T helper 1 cells do is they are going to be stimulated by the production of IL-12, which is best produced. You see this cytokine produced as a result of early immune responses to intracellular threats, intracellular bacteria, viruses that exist intracellularly. A T helper 0 cell that's in an environment with lots of IL-12 will activate a transcription factor called TBET. Activation of TBET leads to the production of Th1 signature cytokines, and this is especially interferon gamma, right? Interferons, antiviral, and IL-2, T cell stimulatory. And so what these cytokines can do is they can activate CD8 positive T cells, NK cells, macrophages. Interferon gamma can also help B cells switch towards antibodies that are better at dealing with intracellular threats like IgG3, which is good at activating complement and good at promoting phagocytosis. All this together, the presence of IL-12 leads to the production of TBET, which leads to these cells producing predominantly interferon gamma and IL-2. And this is what drives a cell-mediated response, which is priming your immune response towards the killing of a cell. And in this case, that's important because the cells are what are harboring the pathogen. And we see that here, right? And this is arguably a pretty nice way to view it. Now, our Th2 cells, this type are going to develop, or these are when Th0 cells are in the presence of lots of IL-4. IL-4 is a cytokine that's produced especially well in early responses to parasitic worms, but also allergens. One of the big producers of IL-4 are mast cells or NKT cells or certain CD4 positive T cells that are activated in the right way. And IL-4 results in the activation of a transcription factor called GATA3. And what GATA3 does is it leads to the production of Th2 signature cytokines, which include IL-4, IL-5, and IL-13. IL-4 and 13 promote B cells to switch from producing IgM to producing IgG4 and IgE, right? And if you ever see IgE, think response to worms or also response to allergens. The production of IL-5 actually activates eosinophils. And so we recall what happens if eosinophils are activated, they're going to express an IgE receptor. They'll bind this IgE, and then when that IgE binds an antigen, we get an allergic response. And so Th2 responses, by virtue of the cytokines produced, really are good at promoting a humoral immune response, which is driving you towards the production of particular types of antibodies. And you can see that laid out quite well here. Now, another major subset is our Th17. The exact composition of the skewing cytokines is a little bit unclear, but we're just going to pull out two that we've already introduced in another chapter, and that's IL-6 and TGF-beta. Both of these are produced early in a response to principally extracellular bacteria, and they're produced by a wide variety of cells. If you recall, IL-6 is one of those common pro-inflammatory cytokines. So if you have Th0 cells that are in this cytokine environment, you're going to see the activation of a transcription factor called ROR gamma T. When ROR gamma T gets activated, it leads to the production of the Th17 signature cytokines, and I've simplified it, so we're just going to worry about the IL-17 family members, principally IL-17A. These IL-17 family members are very pro-inflammatory. They're good at activating innate immune cells. They're also good at leading to the production of more pro-inflammatory cytokines, such as IL-1, IL-6, TNF-alpha. And what all of these help to do is drive especially mucosal immune responses to extracellular bacteria, especially important in controlling those. But Th17 cells, because of their inflammatory nature, also seem to play pathogenic roles in many types of autoimmune or autoinflammatory conditions. You can see that laid out here. Now, the last to talk about with this degree of detail is our Tregs. And there are two variations of T regulatory cells. Their name suggests that they help control something. Our Tregs include, natural Tregs, and these are the ones that we talked about in the previous chapter. Natural Tregs are produced in the thymus as a part of normal T cell development. Then you can have those that are induced T regulatory cells, and these are the ones that are produced during an immune response. And this is when you have lots of TGF-beta. It will drive T helper 0 cells to become these Treg type cells. Now, whether they are natural or induced, for our purposes, we're just going to speak of them consistently. They come from different places, but the remaining events for now are going to be the same. And that is that they will activate transcription factor FOXP3. And this leads to signature features of T regulatory cells, production of IL-10 and TGF-beta, which are more related to anti-inflammatory cytokines. They help turn things down a little bit, but also the expression of things like CD25 and CTLA4 and IL-2 receptor. CTLA4 can help turn cells down. IL-2 receptor can actually bind the IL-2 that's being produced and keep it so that nobody else can see it. And what these do is they help suppress activity of T cell subsets, the differentiation of T cells, among many other anti-immune activities. See that here. Now, there's a cool concept. We're just going to take the very simplified version. What we say, or what's well known, is that, although not exclusively, for the most part, when you're experiencing one type of T helper response, they are cross-inhibited, meaning if you have conditions that are promoting a Th1 response, those conditions block a response or block differentiation towards Th2. And what we consider then is these cells, when they are going towards one subset, they're polarized, meaning they're pushed in one direction, and it's kind of hard to get them to leave that direction. So a very simplified scheme that I didn't feel like taking a picture of the book, so I just typed it out. T helper 1 cytokines, especially interferon gamma, is good at inhibiting differentiation towards Th2 and Th17. T helper 2 cytokines, especially IL-4, help block differentiation help prevent differentiation towards Th1 and Th17. And then Tregs, by virtue of their production of TGF-beta, help prevent skewing towards the other ones. So they just are kind of broad inhibitors of T cell differentiation. Now, there are some other subsets. We will talk about T follicular helper cells in the next few slides. These are a type of T cell that assist in antibody production by B cells. And this is, as you may be shocked to learn, they're going to assist B cell antibody responses towards T-dependent antigens. What's the T that's dependent? It's T follicular helper cells, or TFH. Now, where you find these T helper cells are within the follicle of our secondary lymphoid tissues, and they have distinct characteristics, and I've just listed them for simplicity. They are associated with the transcription factor BCL6. They are related to the expression of chemokine receptor CXCR5, and they also produce IL-21, the cytokine. There are more. You look in the literature, you can find more, but the textbook included it. I think it's useful to know. There are also Th9 cells, and they are so named because their signature cytokine seems to be production of IL-9, among other things, and they seem to play a role in worm responses, some autoimmune conditions, and some cases of chronic inflammation. Now, just a couple of additional notes. Know that this movement between subsets does have a degree of plasticity, meaning it seems like Th1 cells can be shifted into a more Th2 phenotype, and it all depends on the nature of the antigen and the nature of the immune response, what it's going to be. And again, we're just looking at a brief snapshot of the cytokines, just the most important ones, maybe. And so we focus on those predominating cytokines, but know that many of these cytokines that are produced for a particular subset are not necessarily exclusive. In short, it's complicated. So another type of, or besides cytokine production, another function of CD4 positive T cells, by virtue of those T follicular helper cells, is to assist B cells respond to T-dependent antigens. Now just recall that CD4 positive T cells and a B cell are going to do something called cognate interaction. And this is where they're each binding the similar antigen, the same antigen, maybe different parts of it. And what they do is they interact at the T and B cell boundary within the follicle of it. This interaction is necessary because what it will do is it'll drive germinal center formation, which is where we get robust antibody improvements. What are the events? The B cell, using its membrane Ig, so its B cell receptor, is going to bind to this free antigen. What the B cell is going to do is going to bind to this free antigen, and it's going to take it inside the cell. It's going to process that antigen, and then it's going to show it on MHC class 2. So what this means is B cells can act as antigen presenting cells. Who are they going to show that antigen to? Well, MHC class 2 can be recognized by CD4 positive T cells. And it just so happens that the CD4 positive T cells are going to have a specificity towards this antigen, because this T cell was activated within the T cell area by dendritic cells that had processed a similar antigen, or the same antigen. And what this ultimately means is that we are going to have a B cell acting as the antigen presenting cell, and it's going to cause differentiation of the CD4 positive T cell to become a T follicular helper cell. And here we're just looking at a flu protein as our generic, just as our antigen. So a B cell in the B cell area is going to see this antigen. It binds it, it brings it in, it chews it up so that it expresses it on MHC class 2. And then we have a T cell that is ready to activate and differentiate. It's going to bind a specific antigen. It's going to use CD4 as its co-receptor. That'll increase BCL6. What that will do is it'll activate this and differentiate it towards a T follicular helper cell, which is what's going to drive formation of germinal centers. And this is a good excuse to take this slide or this part and tie it back together with our B cell chapter, the events, the activities of the germinal center. Somebody asked, so the B cells become the T follicular helper cells? No, so the B cells act as the antigen presenting cells to cause T cells to become T follicular helper cells. So the series of events for activation are the same as we've already talked about. It just so happens instead of it being a dendritic cell, it's a B cell, right? Yeah, that's a good question, a good clarifying point to make sure we know. Yeah, good. So we have this germinal center form, and this actually is stimulated because T follicular helper cells express a lot of CXCR5 that guides it to form this germinal center. And within the germinal center, we're going to have these interactions between these now differentiated T follicular helper cells and the B cells that are activating. They've seen their antigen. And what happens is we're going to have these T follicular helper cells are going to produce a lot of IL-4 and interferon gamma. These two cytokines help drive class switching and somatic hypermutation. And then T follicular helper cells are especially big producers of IL-21, which helps both the B cells that are rapidly proliferating and the T follicular helper cells survive and proliferate more. This interaction is what drives, and then the cytokines that are produced by these T cells are what drive the events of the germinal center, which is to rapidly mutate and change your antigen binding sites. And it will on the B cell, so it'll be better or worse. And then we'll have affinity maturation so that we get rid of the bad and we only keep the really good binders. And then we promote their class switching so that we get production of IgE or A or E by plasma cells. And similarly, it has an effect on producing particular producers or particular B cell receptor types of memory B cells. And if you look, these events between the specifically activated T follicular helper cell and the antigen specific B cell resemble very closely what we see in just general T cell activation. Now, there's a concept called linked recognition, and this is the idea, this is the theory that it guarantees that a T cell and a B cell will recognize features that are both present on the same structure. So, for example, we only want T cells that can recognize a flu-specific epitope. We only want them interacting with a B cell that has recognized a flu-specific epitope. We don't want a T cell that's specific to flu to interact with a B cell that's specific for measles. And really, this is achieved because of just the nature of B and T cell antigen recognition. Usually, our B cell receptor binds to epitopes that are external, right? Epitopes that are kind of found on the surface or that are free. And if you think about the way B cell receptors are able to bind, they just bind antigen as they see it. So those are the most likely options. T cell receptors can only see antigens that have been processed. And so they're more likely to be epitopes that are internal, right? Because they've been processed. And what that does is it gives you two cells that can see the same antigen, but they're seeing it from different locations, and they can cooperate. There are other ways that this cooperation is ensured that are way beyond our scope. Okay. So we are descending towards the end. So before we get to the very end, or before we finish, what we want to do is talk about CD8 positive T cells. I think they have very cool functions in the way they work. And we're going to just briefly hit on their activation because it shares many similarities, or the intracellular events are the same. And then we'll talk about how they work, how they go about killing cells, and then we'll speak very briefly about other types of T cell like lymphoid related T cells or lymphoid related cells. So CD8 positive T cells, they're going to bind antigen. They're going to do it in a manner that's very similar to what we described with CD4 positive. When they are activated, they're going to lead to the production of cytotoxic effector cells. These are going to kill pathogen infected cells. They're responsible for killing cells that are put in that are not hosts. So transplanted cells or tissues. This is a principle behind graft rejection. They can also kill transformed cells such as malignant or tumor cells. And we term these CD4 positive T cells upon activation killer T cells or cytotoxic T cells. I'll use the abbreviation CTLs. The cell that is being killed is the target. So the target of the CTL will actually could be any cell as long as it is expressing MHC class 1. And that's important because what do we remember about who it is this CD8 co-receptor recognizes, MHC class 1. So when we generate these effector cells, what we're going to focus on is viral infection, but we can translate these to other types of CTL stimulating antigens. So using virus as the example, the process first has to start that antigen has to be observed. And so this could be that the virus gets brought or circulates its way into the draining lymph nodes through the lymph. It could also be that dendritic cells capture it, in the periphery and take it there. Within the lymph node, that virus is going to be eaten by either macrophages or dendritic cells. Somebody's going to take it up. And then the events of CD8 activation are very similar. You have a first signal, which is where we have our T cell receptor recognizing peptide, but in this case, it's going to be presented in MHC1. Also, we need CD8 binding to that MHC1, that co-receptor, to reduce the threshold, to reduce that barrier. We're then going to have a second signal, which is again co-stimulation. CD28 on the T cell is going to interact with B7 family members. CD40 on the T cell is going to interact with CD40 ligand. And then we need a third signal, in air quotes, third signal, and that's just going to be the particular cytokines that are available. These are going to be produced especially by innate immune cells and antigen-presenting cells that have seen this viral threat. IL-12 is especially important. Dendritic cells are an important antigen presenter. They're also they're such so because they are good at presenting antigen. They're also good at secreting IL-12. Additionally, interferon cytokines, interferon alpha and beta, are good at contributing to CTL activation as well. And if you recall, interferon usually relates to antiviral response. So we have a couple of different pathways that can lead to CTL activation. Probably the easiest to consider is the idea that a dendritic cell can directly activate a CTL. And then there are some antigens that require the antigen presenting cell can't directly activate the CTL. They need help from a CD4 positive T cell. So the easy one first, direct activation. A dendritic cell is going to have this antigen and it's going to present it via MHC class 1. This could be because the dendritic cell was infected by the pathogen. It could be because the dendritic cell captured the antigen or recognized it with its pattern recognition receptors and digested it to show it with MHC class 1. Or it could be this event that we didn't talk about because we didn't do that chapter, but it's called cross-presentation. And that is where some macrophages will capture some of these antigens because that's their job, but then they will hand them off to dendritic cells to show them. Right, so that's a thing. And then all of the activating signals in this case are going to be provided by the dendritic cell. They're responsible for the cell-to-cell contact you see. Right, first and second signals. They are also going to be secreting the IL-12, that is that third signal, that cytokine message that helps the cell proliferate and differentiate towards many effector cells. Now, with activation, the CTL itself will start to produce IL-2, which will further enhance its activation and its proliferation, producing these CTL clones, as well as acting in an autocrine and eventually a paracrine manner. And the more complicated setting is that where our CTLs need to be activated with help from a CD4 positive T cell. The necessary events to this are not well understood. So what is sort of understood, or at least speculated, I've just listed here. A dendritic cell will grab an antigen. It's a presenting cell. It'll grab that antigen. It'll process the peptide and it'll express it with MHC class 2. That will activate an antigen specific CD4 positive T cell. It'll produce IL-2. That dendritic cell will also present those same antigens because it captured it. Dendritic cells will just chew up the antigen and express it with anything they have. So it will also be expressing that similar antigen with MHC class 1, which then activates an antigen specific CTL. And the IL-2 that is being produced from the the T helper cell will be the necessary cytokine signal to get the CTL to proliferate and continue its activation. So we actually have this triad, right? We have three cells that are kind of all interacting, forming this little triangle thing. And so those are events that can occur. How much they do so in a physiological setting is unclear. Now, there is another method, and this is where those events actually take place at different times. And this is what is called the licensing pathway. And in this case, it's where you have an immature dendritic cell. It's going to interact with a CD4 positive T cell. That's going to activate the T cell, but it's also going to force maturation, right? The things we talked about at the beginning of this chapter. It's going to cause the maturation of the dendritic cell, and that dendritic cell can produce IL-2. And now this now activated and mature dendritic cell will go and activate CTLs in a manner observed directly, right? So the licensing pathway says that not only is the dendritic cell trying to activate the T cell, but that act tells the dendritic cell, hey, because you activated me, a T helper cell, you yourself are showing the right antigen. Go and show it to the back. Go and show it to the CTLs. Now, once we get intracellularly, all of those steps we learned before, this thing, if you can draw it for a CD4 positive T cell, you can draw it for a CD8 positive T cell. You just need to make a few extracellular, a few antigen recognition changes. You need to make sure that if you are talking about a CTL, you are replacing CD4 with CD8, and you are replacing peptide and MHC class 2 with peptide and MHC class 1. Krista, in the licensing part, this part. Oh, right. And so in this case, it is IL-12. Right? IL-12 is the cytokine that when you have a dendritic cell activating a CTL, if it's producing IL-12, what that will do is it will give the CTL the message it needs to start start producing its own IL-2, which will cause it to proliferate. If these things are working in a triangle, it's going to be both of these interactions occurring simultaneously with the production of IL-2. If it's just a mature dendritic cell interacting with a CTL, you want the production of IL-12. Cool. That's a good question. Good clarifying point. The more you do it, the more you come to realize IL-2 and 12 and all that, but they are so close in name that it certainly can be it can be confusing, even for somebody who's done it, I guess, long, maybe not well, but at least for a long time. All right, so I guess the fun part, that's morbid to say, the killing part. When the CTL is activated, its job is to go and to kill something. So how is it that an activated CTL kills? Well, what it needs to do is it needs to find a target, and that target has to be expressing MHC class 1 with a peptide with this antigen that'll present it. So the CTL is who is going to have the CD8 and the T cell receptor, and it can recognize the MHC class 1 and this peptide on a target. And when that does, not only will you have that interaction, but you will have an equivalent of a synapse form. You're going to have contact between these two cells, because what you need to do is you need to have time for that message to get translated or to get transmitted. And you also need time for the killing events to occur. There are two different ways that cytotoxic T cells kill, and that is to release granules, so they dump this material out that goes and destroys the target cell, or they can stimulate protein interaction on the surface, which also causes the cell to die. Now, once that cell dies, the CTL lets go. It's sent its message, you're dead. It lets go, and then it goes to find the next target cell. And there are fairly cool videos. And if I can find them quickly, what I'll do is I'll find them and I'll share them, because it's kind of cool. You see them interacting, and then one cell kind of withers and the other one carries on its merry way. And those are actually from microscopes, they're watching those cells do their thing. Now, what you've noticed, the activation of a CTL, that occurred through an antigen presenting cell. And then the act of killing is when it sees MHC class 1 on some other target. And so killing and activation are separate events. It would be unfortunate if a dendritic cell was activating a CTL and these events were not separate. What would keep the dendritic cell from getting killed? More complicated. So we have disparate events. You get the CTL activated against a particular antigen. You send those clones out to see that antigen. Now, when we kill, or when these cells kill, the predominant way this occurs is through the release of these CTL granules. Cytotoxic T cells have these granules in their cytoplasm, and when they bind targets, so when their T cell receptor sees sees the specific peptide being expressed by MHC class 1 and all the other events are good, that will induce a response where the cell will let go of its granules, and those granules will fuse with the target and it delivers a load. And the load it delivers is predominantly two things: a molecule called perforin. And perforin looks a lot like perforate. And so what perforin does is it forms these holes in the target cell membrane. Part of these granules are also an enzyme called granzyme. And we have talked about granzyme, or have we? No, maybe we didn't talk about it. But what granzyme does is they just indiscriminately activate proteins, and specifically, the specific proteins that they are good at activating, I should say, are those that are related to apoptosis. And I had a more advanced slide that I took out, and maybe I'll end up putting it back. But perforin forms the holes, which can cause the cell to lyse, coupled together with the fact that granzymes being released and entering in through these holes that are forming, going and turning on apoptosis. So you get this cell that is expressing peptides that are related to the virus. It's going to die. Now, the other way to kill, which is one that gets represented but is not as predominant, is the interaction of Fas ligand and its receptor Fas. Fas ligand is expressed on the cytotoxic T cell, and the binding partner Fas is expressed by the target. We can see this part here. So when Fas ligand interacts with Fas, that sends a message into the target that activates pathways related to apoptosis. So these two things, whether together or separate, get a cell to experience strong signals to die. Cell dies, and the virus that was within it also dies. Now, CTLs can only interact with only interact with and kill cells that are expressing MHC class 1. Generally, only intracellular pathogens make the peptides and get processed the right way to be presented on MHC class 1. And what this does is it helps restrict it so that our cytotoxic killing effects are only going to occur on host cells that are infected, because non-host cells won't express those peptides. So in theory, there's going to be little inappropriate host cell killing. Mostly what it's going to kill is going to be a cell that's expressing these altered peptides. And then we have class restriction, just a reminder that antigen that's captured by antigen presenting cells, that not necessarily an intracellular thing, is processed and presented a little bit differently so that it puts it on MHC class 2 to activate CD4 positive T cells. Now, just a brief word about memory cells, brief as in one slide. Memory cells are produced during T cell activation. They're going to be produced at different checkpoints. Both memory cells related to CD4 positive and CD8 positive responses are produced, and they can be broadly split up into two types. You can have those that are effector memory cells. These are usually these memory cells are found in the tissues, usually related to where the antigen came in, and they're kind of in these tissues so that should that antigen reappear, they can activate very quickly. They're already at the place where the threat might be, and you see that. Before you get to that point, you'll form, which is kind of a backup copy, right? It's not as differentiated. And these are central memory cells. And you find these central memory cells are found in lymphoid tissues, which is a place where antigens might be seen, right? You have your fast responding memory cells in the tissue, and then you have those that take a little bit longer, are a little bit slower to get activated. But when we say slow, we still mean within the course of a few days, not the 7 to 10 days that the normal T cell activation takes. So just 3 slides to finish. What about some of these other subsets that are related to the lymphoid lineage? Natural killer T cells, we've already introduced, so most of this is probably redundant. That's okay. NKT cells express both T cell receptor and molecules such as NK1.1 that are related to natural killer cells. They're principally found in the lamina propria of your intestinal tract. And what they do is they recognize lipid and glycolipid antigens that are related to pathogens, as well as particular glycosylated lipids that are related to host cell membranes. When an NKT cell is activated, they are especially good at secreting Th2 responses, some Th1 cytokines, but they're especially good at producing lots of IL-4. And what this looks like is that NKT cells help drive responses towards a Th2 subset, which is the one that works best in producing antibodies that maybe helps tackle your certain types of bacteria. Gamma delta T cells, which we've talked about before, recall they use the gamma delta T cell receptor chains instead of the normal alpha beta. That has consequences because remember, they don't express co-receptors. They don't see antigen through MHC class 1 or MHC class 1 or 2. You predominantly find these gamma deltas in barrier sites such as mucosa and skin. And they have features that are both adaptive and innate. And they are recognizing a group of antigens called phosphate antigens that are expressed and shown by a particular protein, which is CD1. In a previous chapter, we were more specific with CD1D. It's okay to know CD1. And once they're activated, they have both cytotoxic ability and the ability to secrete cytokines, especially inflammatory cytokines such as TNF and interferon gamma. And they play early roles in responses. And they also help probably clear some of these self-antigens and clear altered or damaged host cells. And the last is just the briefest of words. This is the idea of an innate lymphoid cell. And these ILCs are truly innate cells, but they come out of the lymphoid lineage. They do not express T cell receptor or any antigen-specific receptors. They will express pattern recognition receptors, right? They're innate cells. And they're grouped into 3 classes that are fairly analogous to what you see with your T cell subsets. It's based on what molecules they express on the surface and what kind of cytokines they produce when they're activated. ILCs seem to be most associated with your mucosa. And one that we've talked about, maybe the best known member, depending on the text and the group, how it gets done, natural killer cells are considered an ILC1 member. Right? Natural killer cells are innate. So with that, we have finished. I think it seems like there's a lot of information here, and I guess there is, but a lot of it is going to be redundant to itself. You're going to have lots of repetition. And so as you're kind of going through and putting this in your mind, as with other things, organize it, know, oh, okay, well, this CD molecule is related to this basket. And so if you know who belongs in which place, you can then start to shape what are the events that need to occur. Especially useful is to be able to, probably could have just gone through it anyway, is to be able to sketch out those those signaling events. Recall on a test if I ask you to do that, or if that's a part of your answer, you are always welcome to draw that out. And as long as you send me an email of anything that you want considered, whatever the time frame is, I forget. So being able to draw it can help, or arrows or whatever works for you. So with that, I'm going to stop sharing and I'm going to stop.
Recording stopped.
Okay. With that, we are done with Unit 3. That also means next week on Tuesday, we have presentations who are going to do. We are also going to, I'll